mess with your heads. Let's see, my favorites are those guys. So what happens if I change I'm going to change two positions. Doesn't really matter, I can change two positions. I'm going to change UCC to AGC. UCC to AGC. What do we got? It's a silent mutation. What's happening? We're going from a serine codon on the top, right, to a serine codon over on the lower right. Serine to serine. You got two mutations in your DNA. The protein doesn't care. But think about this. The protein might not care. But what if we started with what? UCC? What if that UCC was an important component of the 5' exon-intron boundary, such that the splice site is no longer recognized properly, and then you get screwed up splicing, right? So those sort of things can also happen. So the mutation in principle of the exons were put together properly wouldn't make any difference. But what if it screwed up the splice site, so now that splicing is wrong, and maybe now the whole frame of the thing is off? All of those sorts of mutations can happen at one time. So the last one that we need to deal with is, let's go with a CCC to a GCC. We're going from a CCC to a GCC. Right? What are we starting with? What are we starting with?
Proline.
We're starting with proline, CCC. We're going to a GCC. What do we got now? Alanine. And what's the problem? Missense mutation. Chocolate or non-chocolate? I'm only going to bring chocolate from there. Whoa! I have to throw over. Does that work? Does everybody understand that general principle? You're going to have to follow those through on your exam and deal with different things. Does that work? Now, we do have a problem that gentleman back there asked, and other people can ask questions. You got a question?
Yeah.
What?
So, we have like an insertion that leads to like a nonsense mutation?
Absolutely. You can have an insertion, a frame shift mutation, and as you start translating in the new frame, you will more commonly than not run into a nonsense code. That will happen. But the mutation is deemed a frame shift mutation, and whatever, because a frame shift mutation is undoubtedly probably going to lead to multiple missense mutations, potential silent mutations downstream of it, and it usually terminates in a nonsense mutation. Yes, ma'am?
If there is a problem with the splicing as a result of a silent mutation, is it still considered a silent mutation even though overall there's a problem?
You get into a gray area there of what it would actually be termed. The mutation itself, if you consider the protein, would be a silent mutation. How it would be reflected based on it interfering with splicing would depend on how it interfered with splicing. Because maybe splicing occurs so that that whole exon is included in the final transcript, but it eventually screws up some downstream event where there would now be a frame shift or a nonsense. So it's hard to describe. So those are the mutations that we have to deal with, right? So probably, is there any concern with them? Yes, sir?
So if there's a mutation and a wrong amino acid is added, are there any repair mechanisms to fix that, or is the whole polypeptide chain is trash?
I'll have to answer each part separately. So for those of you who didn't hear the question is, if there's a missense mutation and the ribosome translates that sequence, the first part of your question is absolutely true. There will be a wrong amino acid at a particular position. Position 47 of our protein was supposed to be a proline, and that position is now an alanine. Can it be repaired? There are no known mechanisms inside the cell to remove one amino acid and put a new amino acid in its place. Because how would the cell know which amino acid to put there? How would it even know that that amino acid is wrong? Right? There are no mechanisms to selectively remove an amino acid and put in a new amino acid. Now, amino acids are modified. They are phosphorylated. There are different activities that can be attached, methyl groups and other things can be added to amino acids. But in terms of whole scale, cutting one out and putting a new one in, no. The second part of your query was, I believe it was termed, is that whole protein trash, right? It depends, is the long answer. Again, if we go back to the synthetase, that big blue blob is probably 500 amino acids long. Some of those amino acids are critical for adopting the exact blue blob shape that it is. Some of the amino acids are largely unimportant for that. Some of the amino acids are critically important for radically recognizing the tRNA in its proper form and adenylating it and getting it all going, right? And some of them aren't. What really messes up a cell is not so often when they make a piece of trash, but when they make something that still folds up. But imagine you're supposed to be making a tRNA synthetase that takes tryptophan and puts it on the right tRNA. And now you've made an enzyme that's a little bit tweaked. It's a synthetase. It can still do its job of taking an amino acid and putting it on a tRNA. But now it either recognizes the wrong tRNA or the wrong amino acid. And it starts mischarging amino acids onto tRNAs. So now your one piece of trash protein is actually creating more errors in the system. And that builds upon itself like a snowball going down a hill, because then you're going to end up with more proteins that have problems in it. And so one mistake leads to another, and you end up just going down a bad path. There are mechanisms in the cell to, and we don't really go into them that much in this class, but we've talked about the half-life of mRNAs. They only last a few seconds. You want to accurately translate the thing, make a burst of protein, and then decide and recalibrate. Do I need more of that protein? Do I need to move on and make a different protein, sort of thing? Proteins are generally longer lived than RNAs, but still there come certain situations where you don't need that protein, right? In most humans, for instance, we need an enzyme that can digest milk when we're nursing. And the vast majority of us should become deficient in that protein in adulthood. Now, most of us still drink milk, but it's actually kind of weird if you think about it. No one goes out into a field and like attacks a cow and gets the milk directly from it. It seems very sterile to drink it in a bottle in a store or something like that. But in reality, we're not designed to drink milk, and most of us should get rid of that protein. And so there are mechanisms to clear that protein from our system to make sure that the encoding gene is not even transcribed anymore so that we don't have any capacity to make those proteins. Does that make sense? Other queries, questions? I'm happy to answer questions. Those are the best way to get through class. They really are. Yes, ma'am?
Is that how you become lactose intolerant?
Absolutely, that's how you become lactose intolerant. And you're supposed to be lactose intolerant. Don't think of it as, I'm trying to get to you. Don't worry. It's going to be crazy. I'm going to probably hurt myself. But that was a winner. I call that. I expect applause when I learn that. That's the end of the chocolate peak. You have to eat leftover Halloween candy for my kids. Other queries? All right, here's the biggest problem. These things are really important. This is a slightly different answer to your question, but it's the only one I have a slide for. So the question was, what happens if you take a stop codon and make it into a coded codon?